Our neurons are electric, and this bioelectricity is fundamental for their development, for the formation of networks, and above all for communication. In short, electricity is essential if our brain is to function. Just as with the appliances we plug in at home, electric current flows in our neurons in the form of ionic currents. That could be calcium, potassium, chlorine, or sodium atoms in which there are missing or extra electrons. This neuronal current doesn't flow freely. It's controlled by channels, which are very special proteins found in neuron walls, and these allow some ions through if very specific criteria are met. There are many types of channels which are regulated by many different processes, creating a wonderful complexity that allows neurons to create a complicated language that's based on electrical signals. Understanding how these channels work at the molecular level is fascinating, and it's the fundamental basis of brain function and of the work that we do in our laboratory. By using systems from molecular biology, genetics, and electrophysiology, as well as advanced methods of molecular imaging, we're able to study a channel's molecular movements in perfect detail and see, for example, how these movements are affected in diseases like epilepsy, migraine, and muscular dystrophy when these channels fail. If neurons are so small that we can only see them through a microscope, the channels are even smaller, about 1/1000th of a millimeter, and they can't be seen with a conventional microscope, but there is a technique to measure the electrical current they produce. This technique, known as patch clamp, for which doctors Nair and Sackman received the Nobel Prize in 1991, is a standard method in our laboratory. But there is still more that we can do. In addition to measuring the electricity that the channels allow to pass, they can also be lit up. There are luminous proteins from marine organisms which can be adapted to the channels in order to allow us to understand how these incredible molecular machines work. Although they are luminous, we can only see them on a large scale, but the light still gives us information about what the channel is doing, changing its qualities. For example, when someone holds a torch in a darkened room and moves it, they create a shadow and changes in light. This helps us to see where the torch is, or even what type of movement is being made. Although we don't see the person holding it, we don't know if it's a man or a woman, for example, we can still obtain plenty of information about the person without actually seeing them. In the same way, we have managed to deduce the molecular movements of a channel while it's functioning in the cell. One of our most recent studies involved lighting up a channel that was implicated in epilepsy by implementing a complex technique combining current measurements with the light technique in order to see how this very complicated machine works. This study allowed us to see that when the calcium that we have in our cells increases, it joins a particular part of the large channel protein and produces a very big movement, at the scale that we are using, of course, which operates mechanically, like a molecular lever, to make the channel's door open and let the potassium ions through, which in this case are the ones that produce the ionic current in the neuron. What would happen if the lever were to break, as in the case when the channel is pharmacologically or genetically damaged? What happens is this calcium effect doesn't open the door, and as a result there's no signal. If there's no signal, the neural language is incomplete, and then you get disease. Our work is possible by combining many techniques. We need to work with animal models from which we obtain the cells that we work with. We use molecular and genetic biology techniques to obtain channels in preparations that can be registered with our imaging and electrophysiological measurement systems, or observed under the microscope. Our results are quantified and analysed carefully and rigorously. The brain is one of the great unknowns of the 21st century, and it will probably continue to be so in the next century too. With different levels of complexity, a highly complicated molecular system makes it possible for us to feel, to arrive at opinions, to have doubts, or to remember. In all of these processes, the ionic channels, of which there are more than 200 types, are fundamental. If we were able to understand how they work at the molecular level, we would be able to know how to fix them if they fail, how to improve them if they don't work well, or how to optimize their functioning if this would improve our lives. The unknown aspects of the brain must be approached at all levels, both holistically and from the perspective of its essential parts. We are just one of many groups that provides new information in order to be able to understand how one of the most wonderful and one of the most mysterious systems evolution has brought us actually works. In the study of the brain and neuroscience, we are actually experiencing a breakthrough. Although we said before that we can't see the processes that are very small or very fast in the cell, I must admit that it's now changing at a very rapid rate. The scientific community is developing super-resolution systems in which we can see processes that occur in the neuron, something that was thought impossible until now. We are part of this revolution, and we want to contribute with the description of molecular processes that occur in the zone through which one neuron communicates with another, in zones as small as 1/1,000,000th of a millimeter, and at speeds like 1/1,000,000th of a second. In our brain, this happens continuously and on a massive scale, and it's now within our reach to understand how this fascinating process takes place.